And we are going to talk about block copolymer and homopolymer blends, and specifically we're going to look at micelle formation. So the scenario that we're dealing with here, I'm going to finally, so I have my A block, I have, actually let me be consistent with our figure below here, so make sure that I want to copy these guys, so A and B, so that's actually my B block. I'm going to get my A block right here, so A, this is A, and this is B. And we're going to be in basically a situation where we have lots of homopolymer A. So we're mixing dye blocks in this bath, essentially, or in a solvent. You could kind of think of it, solvent of A homopolymer, as you can see here. And we're going to see when micelles form. So micelles are basically the spherical aggregate of dye blocks. You're going to have this core region, which and this corona region. So the corona region exposed to the solvent. So if we were to kind of self-assemble here, so in the core, again, it's called corona and core because it looks like an eyeball almost. So I'm going to have so here, so this is going to be inside of here. This is my core. And then I'm going to have another scenario, a little range here. This is going to be my corona. And then my solvent is going to be just kind of around here. Why does the A and B, why does it kind of self-assemble like this? Well, we know that chi AB, that my chi AB is greater than 0, and we know that chi AA is equal to 0. So our A block wants to interact with other homopolymer A, and our B block does not want to interact with that solvent whatsoever. So it wants to shield, basically, the corona shields the core from these unfavorable interactions. And there's going to be a specific concentration of dye blocks, so a concentration, a number of dye blocks, where micelles will start to form. And again, what do you think the energy competition is going to be? You guessed it, entropy versus enthalpy. But also, since dye blocks are large and thus our mixing entropy is very, very, very small in terms of magnitude, our critical micelle concentration, the concentration where this occurs, where micelles first form, is going to be pretty small. But we'll see that in just a second. And it'll be a nice, again, fun derivation. So we're going to be mixing homopolymers, and then we can kind of see what we're going to be dealing with here. So micelle formation. So spherical self-assembly of molecules, again, self-assembly, or essentially self-assembly here. One reagent prefers to mix the solvent, one reagent tends to avoid. So we're going to be looking at specific examples of dye blocks, so dye block copolymers A and B, but this is a very general behavior, and specifically one very, very important. So the core shields it from, or the corona shields the core from our solvent, the corona mixes with the solvent. That's kind of the key aspect here. Corona mixes with solvent, core hides from the solvent. So you've all hopefully encountered one of these systems before that forms kind of this micelle or this self-assembly, which is lipids. So lipids are amphiphilic. They have a hydrophilic head, hydrophobic tail, and your lipid bilayer self-assembles. Hopefully, even if you're a mechanical engineer or material scientist, you've run into lipid bilayers. So you have kind of your water molecules, your solvent around here. So we want to have our hydrophilic heads be exposed to water, hydrophobic, hide them. So that's kind of our example. Again, I really like this figure here from your micelle. This is exactly what we're going to be kind of seeing. So core, corona, looks like that eyeball with really red eyes. So that's what we're going to be working with here. So, excuse me, at a specific concentration, CMC, ordering will form this kind of micelle structure to reduce those number of unfavorable interactions. And basically, you have the lipid heads facing outwards, hydrophobic tails inwards towards the core, again, to hide those interactions. And you could form even more ordered phases coming up too. So this whole dye block homopolymer mixture is going to be very, very similar. So homopolymer solvent, dye block is our solute. We're going to have the core being the other polymer that's not similar to the homopolymer. And at some critical concentration, that critical micelle concentration, the dye block copolymers will aggregate because there's too many interactions. So you can imagine if I have one dye block and then there's just solvent, solvent, there's not too many unfavorable AB interactions. But as I start to increase the number of my dye blocks in my system, there's too many unfavorable chi AB interactions. So it will want to, again, to minimize this, it will want to spontaneously self-assemble. Even though we lose delta S of mixing, we know delta S of mixing is small, it will want to self-assemble to reduce the number of these unfavorable chi-AB interactions. So that is essentially what's going to be happening, and we're going to derive that expression in that next video. So again, minimize total amount of unfavorable interactions with the solvent. That's kind of the driving force here. So competition, enthalpy, and entropy. Cost entropy, because we're going to order the die blocks. They're going to probably stretch or compress again. We're not sure. We're going to keep it general, but we're going to save in enthalpy, minimizing those bad AB interactions. So we're going to put those B blocks basically in the core of our micelle. So the enthalpic term, again, is going to be our interfacial energy. Driving force is enthalpic, and again, we're going to have to, but the big thing here is we're going to have to consider this influence of the free homopolymer system. And we're going to have to do some, again, some derivations, but also some change in variables and notation. So more on that to come in the next video, but that's kind of, again, your qualitative understanding of how we're going to deal with or visualize or conceptualize this critical micelle concentration. So I'll see you all in the next video. Have a good one. Thanks.